to the study of cultural heritage artifacts, so old things in general. Um, the cut to the chase. What was I doing in Durham? So the group that I was working there is sort of they call themselves Team Pigment, which I think is really funny. But um, about a year ago, they published this book, and it's a tome. It's like, an inch and a half thick. Um, they collected data on over 300 manuscripts spanning about 1,000 years, studying the pigments that were used in the illuminations of all these different manuscripts, right? And trying to understand sort of the time and space by which various pigments propagated from Europe or Asia into England to be used in these manuscripts. But they did this through essentially point measurements. My colleague, Andy Beebe, is a spectroscopist extraordinaire. So he understands all things about point spectroscopy, from fluorescent spectroscopy to Raman spectroscopy to reflectance spectroscopy. And Richard Gamison is one of the world's best paleographers. In other words, he can look at a medieval manuscript and tell you all kinds of things about it just by the way it was written, not necessarily by what's inside it. So they've been doing this work for about 10 years with point measurements. They had a multispectral imaging system, and they pretty much only used it to make pretty pictures to go in the book. And then about a year and a half ago, they acquired two hyperspectral imaging systems, a Vis-NIR system and a SWIR system. And they didn't really know what to do with it, right? Because Andy's a point spectroscopy guy and Richard's a paleographer. So my job there was to extend all of the work that they've been doing to image-based analysis. So none of what you're going to see here is rocket science. If you're at all familiar with how we process hyperspectral and multispectral data, all of this will basically be very, very familiar. But essentially what we did was we built an end-to-end workflow that goes from collected raw data to calibrated images to essentially information products, whether they be pigment maps or enhanced contrast or all kinds of different things, 3D renderings, all kinds of stuff. So Izzy Moyer is in the crowd over there. What do we do with these things? So we build multispectral and hyperspectral images and deploy them in libraries, archives, museums, conservation studios, all kinds of things for a few main goals. Preservation and conservation is sort of the primary thing. We're trying to preserve these artifacts and in some cases inform the conservation scientists about the materials that are actually used so that they can do their work in conserving the artifacts. A lot of it's about discovery, right? We're trying to learn more about the artifact itself. And sometimes because we're making digital versions of these things, even though we are not doing digitization, we can produce digital versions of these things that can be used in exhibits and publications. And so it provides more access to people to get to these sorts of things. We primarily use multispectral and hyperspectral imaging. I'm not going to talk about it, but this is our little Misha system that my colleague Julie Decker, who's in the crowd, and Roger and I built and designed under an NEH grant. And this is Izzy last year. We won a small grant from the Ministry of Culture from the government of Croatia to put one of these things in the National Archives in Dubrovnik, Croatia. And now Izzy is actually leaving on Monday to go there on a Fulbright scholar to work with it. So what are we trying to learn? So these are my sort of partners in crime. This is Team Pigment. So in the foreground is Andy Beebe, the spectroscopist, and in the background is Richard Gamison, the paleographer. And they like to joke how I don't meet the physical requirements for joining Team Pigment because I still have most of my hair. But anyway, this word, codicology. Codicology is a cool word. It comes out of like anthropology, archaeology. It's the study of the artifact as an object, not necessarily what the content of it is, but how was it made? What were the materials? What is the context of the object about when and where it was made and created and why and all those sorts of things. So, we started out doing a lot of this stuff and we still do a lot of work on faded text. So text that has either been erased intentionally or just faded over time or is unreadable because of water damage or something like that. This work, obviously, they're called Team Pigment for a reason. They care about the pigments in the illuminations. So can we map out the pigments that we can use for this? But in general, we try to help scholars both understand the content of these artifacts as well as the context, right? How were they made? Why were they made? All that sort of stuff. This is a real simple example that requires absolutely no image processing whatsoever, but it sort of indicates why we do it. So this is, again, part of the Guff map. This is a zoom in on an area of Scotland with a giant wolf or a hound here. Again, this map is from around 1400s. It lives in the Bodleian Library. So this is what it looks like in RGB, right? If so, you just walk up to this thing and you look at it, this is what it looks like. All you have to do is image it in the infrared and you get something that looks like this, right? And so this immediately tells us something about the pigments that were used for this thing. The reds here that disappear are vermilion, which is transparent in the infrared. Most of the drawings that are down here and around the outline of the dog, they disappear because they're iron gall ink, not carbon black ink, right? And so we haven't done anything to this image. All we've done is we've collected imagery in the visible and imagery in the infrared. And just by making a false color in the infrared, we can already start to tell you about the materials that were used to make this particular object. So like I said, we primarily work with multispectral and hyperspectral imaging systems. This is the Durham University setup from last winter. It hasn't changed too much. It's bright in here, so it's hard to tell. This is at the reading room at the University of Edinburgh. So there's a prosumer DSLR up here. So a very expensive, very nice Canon camera with a very nice lens that has had the Bayer filter and the IR filters taken off of it, right? So it's just got a straight silicon detector. That turns out to be very expensive to do. So that camera system is $20,000 or $30,000 just to buy the camera and the lens. We do it in a darkened environment. And so we use LED lights, right? Colored LEDs on these various panels. And sometimes we use a spectral filter wheel. So there's actually a wheel right here in front. So essentially we turn off the lights in the room. We turn on a specific LED or we turn on a white LED. We put the filter wheel in the position it wants to be in. And then we sort of collect an image and we just kind of keep going. It's all on a movable frame. I'll talk about that in a little bit. And the artifact goes underneath here with the calibration target. So it's pretty portable. It's pretty easy to take up and set down. It's all run by a little computer here with an interface. So this is sort of what it looks like underneath when we actually set the thing up. We try very hard not to touch the artifacts. These things are very old and very fragile and in some cases priceless. We typically support the manuscript somehow using essentially pillows or styrofoam. And then this thing is a weighted rope like you might put in the bottom of your curtain so they don't move around to hold the pages back. We're trying to always get shadows out of the way. This particular thing is very challenging because it's got a lot of gold leaf on it, which is highly specular. And so that impacts your ability to process the data later on. It screws up the statistics. There's calibration targets in the scene here, both a spectral on step wedge as well as a color checker. And this is basically what it looks like for us to image. And then this is what the image processing, the time it takes to actually do it, right? So we'll actually illuminate, move the filter wheel, take a picture, change the LED, move the filter wheel, take a picture. Don't bump the table because we don't want to have to register the images. In this particular case, we're collecting 16 different images. Each one's recorded as a TIFF. The current system collects about 20 images. Again, we don't want to have to go through all that issue of having to register them. So we just try to, okay, everybody just stay away. Don't jump up and down. This is what some people would call a division of time approach to collecting spectral imagery. In other words, we're collecting them over time as opposed to a division of space or something. And in this case, now we're down into the infrared and the ultraviolet illumination. So we actually image in the UV. So we illuminate in the UV. That induces fluorescence in the parchment because it's an organic material. And a lot of times the ink has sunk into the parchment. And even if it's been erased or scraped off the surface, that suppresses the fluorescent signature. And so you can actually enhance the faded text sometimes by illuminating in the UV and imaging in the visible. And then we go out to the infrared as well for things like that funny example of the dog I showed you so that we can do material identification that way. Commercial HSI is becoming a bigger and bigger thing. So this is a picture of our colleague and good friend, David Howell, who got me involved in all this with the help of Roger. David was a chemist. He's retired now. He's a chemist by training. His specialty was conservation of tapestries. But he ended up being head of heritage science at the Bodleian Library at Oxford. He wrote an internal grant to acquire the Headwall hyperspectral imaging system, a Vis-NIR system. We have a very similar one upstairs in the drone lab right now. And Headwall sort of built this thing. It's a push broom system, right? So you have to have relative motion between the scene and the imaging system. On an airplane, that's easy because the airplane's moving. But in this case, we put the artifact on this translation stage and it slides underneath it. These are good cameras. They're very capable. They're relatively inexpensive, relatively in the sense that, $30,000 or $40,000, but that price is always going down. They have pretty good SNR, pretty good QE, all that sort of stuff. You can get them in Vis-NIR and SWIR, and they're now starting to be deployed in many more lab contexts. David was really the first person to put one of these in a library and in a conservation lab in a library. So he was really a pioneer there. That system is still at Oxford, and it's still occasionally used. This is the hyperspectral setup that the folks at Durham created. So again, they got a sizable donation from an alumni, and then they leveraged that to get a very large grant from UK Arts and Heritage Research Council. So this is the lab, one of the labs that we have. It's literally a renovated dungeon in the basement of the Palace Green Library, which is like a 16th century book, or a building. So it's a cool office, but it literally, you have to go down all these weird stairs and stuff to get to it. Um, there are other systems like this. The National Gallery of Art is working very hard to sort of lead the charge on how you do this in the art conservation world. They have some really, really fancy toys. The Getty Museum, some of you know Olivia Cusio, who just finished her PhD in color science about a year and a half ago. She's leading up the hyperspectral effort at the Getty Conservation Institute right now. The Rijksmuseum in Amsterdam has some extensive collection capabilities and analysis capabilities. Hampton Court Palace is, or Hampton Court, which is part of Historic Royal Palaces, which was the home to Henry VIII. So they have hyperspectral imaging there to study tapestries. And there's a few other places out there. The Louvre has one of these things, a couple other places, but we're still sort of at the beginning of this stuff. This is what the system is. So we've got two cameras here. We've got the Vis-NIR camera and the SWIR camera, and they're aligned, but their fields of view don't overlap. Right? Now, these are line scanning systems, so they're only imaging one line at a time, and then the object moves underneath them. This is the illuminator. It's this very fancy mirror shape, so it only illuminates a stripe underneath the cameras. So what we actually do is we'll align the stripe with one of the fields of view of the cameras, move the object underneath it, tilt the light a little bit so that the illumination spot is in line underneath the other camera, run it again, and that way we get two cubes, a Vis-NIR cube and a SWIR cube. That's a challenge that I'll talk about a little bit later on because the two cubes are not at the same spatial resolution or sampling. They differ by about a factor of 2.3. However, they do have some overlap in their spectral coverage, which allows us to do pretty accurate registration. Now we're working on doing, instead of just resampling and registering them, we're actually working on sharpening the images as well. So what do we do? Why would you use one or the other? First, you have to ask yourself, what is hyperspectral versus multispectral? My personal opinion is how you separate the data out in colors. If you put some sort of a dispersion element like a prism or a diffraction grating in the optical path, then I would call that a hyperspectral system. It would be inconvenient and expensive, but I could build a filtered system with hundreds of spectral filters and collect a data cube with hundreds of spectral bands, but personally, I wouldn't call that hyperspectral. It doesn't really matter. It all depends upon what you're trying to do. Right? Are we trying to simply enhance features on the artifact for visual interpretation? Right? Am I trying to enhance the low contrast text or identify things that have been erased or underdrawings and paintings and things like that? Or am I actually trying to do accurate material identification, quantification, all that sort of stuff, right? And so this is more like when we're trying to present results to scholars who want to read the text or to art historians who want to understand the art construction process. This is when we're trying to work with typically conservators, right? People that actually need to understand the actual pigments and the materials that were actually used so that they can go in and do their job. And in the end, it all depends upon what the institution is trying to do, right? So we don't walk into some place and just say, hey, I have a hyperspectral camera, let's use that, right? We try to talk to the people that are actually doing the other work and say, what are you trying to do? What's your end goal? A lot of times hyperspectral is not the right tool for that. It's way overkill. It's way too complicated and the spatial resolution is not as good. So we really try to work with folks to try to understand what are the questions they're trying to answer. And then we go back and we try to figure out what are the tools that we're going to use. On the processing side, again, we do two different types of things. We do some qualitative stuff and we do some quantitative stuff, right? So there's a lot of traditional algorithms out there that can leverage both the spatial and the spectral information, right? Sharpening, principal components analysis, false color creation, all these sorts of standard things that the human visual system can then use to key off of and identify features in the artifacts, right? If we're going to do quantitative approaches, then we have to get a little bit fancier, right? I've got this data in either 10 or 20 dimensions or hundreds and hundreds of dimensions. I have to come up with a mathematical model for it, whether it's a statistical model or a geometrical model or a manifold model or something like that. And then I've got to leverage that model to actually go in and try to do things to the data and extract information. Um, Typically, when we do this, one of the great failings, I would guess, of hyperspectral processing is that we ignore the fact that it's an image. We tend to not treat the fact that pixels sit side by side, and that's generally because they're at relatively low spatial resolution, so there isn't as much spatial information there. And the algorithms are based on high dimensional statistics or geometry, both linear and nonlinear concepts. And I'll talk a little bit about that, but I'm not going to go into any of the mathematical detail behind any of the algorithms here. So spectral imaging is sort of in general used because it can leverage material differences, sometimes very subtle material differences that are not necessarily visible to the naked eye. That's just in general why we would do this sort of stuff. And so we want to extract information out of both the spatial and the spectral. We want the per pixel signatures are indicative of the material in that pixel. And then the spatial relationships tell us something about, the overall how the thing was made or the context of how it was made or why it was made or things like that, right? In here, right, I'm generally focused on pigment mapping, but because we have a lot of capabilities, both here at RIT and with the folks at Durham, we get asked to do a lot of other things, and I'll show some examples of that as well. But we're generally doing both quantitative and qualitative analysis, right? When we're doing pigment mapping, we're basically want to produce a map that describes for every pixel in the image, what's the label I've given to it, or what's the multiple of labels that I've given it to. It's made up of n of these pixels and these or any of n of these pigments in these proportions, that sort of stuff. So what did I do? So while I was there, like I said, they had point measurement capabilities and thousands of point measurements stored on a server somewhere with very little metadata. And then they had a couple of camera systems. They had the Vis-NIR-SWIR hyperspectral systems. They had honestly only ever used them maybe half a dozen times on things just to make sure they worked. And then they went in the visualizer that comes with the Headwall software, and you can poke a pixel and it'll give you the spectrum. And that was pretty much what they did. On the multispectral side, they had this multispectral camera system that was built in-house by Andy. And he had a bunch of what I would say, I wouldn't even call them MATLAB scripts. They were more like lines of things you would type into MATLAB to do something to the data, right? And they were jotted down on pieces of paper that were all scattered all over Andy's desk. And so they had no workflow, right? And so what I did is I wrote a lot of Python code to build a workflow, right? So on the multispectral side, again, we're trying to get to mostly qualitative results. So the data come off the Canon camera in their Canon RAW 3 format. So we have to convert those to TIFFs. And then you have to take all those TIFFs and you have to build them into a cube that's in a specific format. The NV format is what we use. We want to do flat fielding. We put calibration targets in there. So you go in and you pick out points on the calibration targets and you get a black and you get a white and you do all that sort of stuff. And then you store the data and you get a calibrated multispectral cube. And then we can do processing on it, right? Simple PCA, false color pigment mapping. I'll show you some examples of that. Faded text enhancement, all this sort of stuff, right? So this is pretty straightforward. But what it allows them to do now is when they collect a multispectral image, I can just pull the image, the raw images straight off the camera, essentially press a button and get through this workflow to get a calibrated cube that we can then do things to. On the hyperspectral side, it's a little more complicated. Again, we have two cameras. They are not co-bore sighted and the fields of view are not sort of overlapping and they have different spatial resolutions. So you get these two specific cubes. You can spatially register them. It's pretty easy to do. You can resample and just do a simple warping registration. Spectral splicing means that they have an overlap area in their spectral response functions, and they're pretty well calibrated, but they don't always line up. And so you have to do some things to try to get the spectra so that they actually line up and are relatively continuous. And then we build a full spectrum cube. We don't have to do any radiometric calibration here. That's all done internal to the software on the Headwall side. We image, there's a spectral on strip. We collect a dark image. We image the spectral on strip. For a white, we use all that to get to estimated reflectance. So now I've got an image. I put it over here. I want to do processing at the per pixel level. I want to go in and either label it, think of it like a classification problem, or I want to do per pixel unmixing. So we can either extract end members, which are sort of hyperspectral lingo for unique signatures that you find in the scene, or I can get them from those thousands of FORS measurements that we've taken offline. I can build them into a spectral library. Now I can try to do material identification at the per pixel level. I'm going to show you an example where we also have the capability to image these things in 3 dimensions, and then we can register everything together. And it turns out you can learn a lot more about how these manuscripts were made if you can actually view these information products that come out of this along with the 3D structure of the object. Here's just a little bit more detail. Oh, I should say that we put 2 papers into the SPIE conference in the spring on this half, one by Jose Macalintal, who's a PhD student working with me on this, and one by Amir Hassanzadeh, who's a staff member here, working on the spatial registration and the resampling. And now we're working on hyper sharpening of the SWIR cube, so you get better radiometry as opposed to just resampling it. The registration and spectral slicing is pretty straightforward, right? I'm going to get control points either manually or automatically, and then I warp the images. We don't have big registration problems because I'm not moving the object and the camera systems are right side by side. And so getting them registered is a pretty light lift here. Getting the spectrum right is a little bit more complicated because we sometimes get really glinty materials and things like that. But we can get to a pretty good registered full spectrum cube. Once I've got that, again, I'm going to do end member extraction or I'm going to use an offline library. I put these into here. Now, we tend to do things in this building in reflectance space, right? Chemists don't like to do that. They like to do it in this other space called the attenuation space. And it's a nonlinear transformation of the data. When you've got these intimate mixtures that have a lot of internal scattering and stuff like that for point spectroscopy people, they do this data transformation to get it into the attenuation space where different materials can be slightly more linearly separable, right? And so we have the ability to do that. We haven't encountered a lot of cases where we've had to do that yet, just because when they've made these medieval manuscripts and some of the maps that we've looked at, they don't do a lot of pigment mixing. They have everything is either red or blue or yellow or green or something like that, right? But we have the ability to do that. So we can also do things multimodally. You're going to see this manuscript a lot. This is essentially our test object. It's a 12th century manuscript written by this dude named Simeon. And it's in the library at Durham, so it's really easy for us to get access to. We do a lot of point measurements, so Raman spectroscopy, X-ray fluorescence. We can actually do imaging in X-ray fluorescence at really low spatial resolution. And then fiber optic reflectance spectroscopy. Andy built his own FORS system because the standard ASD system doesn't have quite the spectral resolution that we need. And then on the imaging side, we can do 3D scanning using photogrammetric stereo with this very expensive and fancy system called a Selene. We can do multispectral and we can do HSI, right? And other than the HSI system, everything has to be portable. You have to be able to take these things to the manuscripts. The manuscripts will not come to you, right? So all of this equipment, other than the HSI is in principle portable, right? But you have to take it apart and it's complicated, so we don't do it. So this is me standing at the train station in Durham on our way up to Edinburgh to image a bunch of manuscripts. So this is that Simeon manuscript. This is just an example of the types of things that we can do. So Simeon was a cleric and a scribe who was trained in Normandy, but he was brought to Durham in the 12th century to write a book on the history of the church in Durham. One of the sort of famous things about Durham is there's an 11th century cathedral there. It's one of the largest cathedrals in England, and it holds the relics of lots of really old saints, Bede and Cuthbert and all kinds of those people. So it was a really, really influential place in the early 12th, 13th, 14th century Christianization of England. Anyway, he did things a little differently because he came from Normandy. He wasn't in England. So this is an interesting example for us to study because the techniques and tools and things like that he used were different. So the question is, again, we don't care about the text here. Up here in this illumination, which is a capital T, can we identify the pigments and materials and maybe the tools that were used to create this thing? So we imaged the heck out of it with everything we have, 3D, HSI, MSI, XRF, point spectroscopy, all that sort of stuff. When I went to do the unmixing results, the end member analysis, this was a non-expert. I went into here and looked at this and said, there's a couple of types of blues, a couple of types of reds, a couple of types of greens, some whites, some browns, some blacks, and I just pulled spectra by hand, which is not the right way to do it, but we're more just demonstrating the workflow. So XRF, X-ray fluorescent, tells you about the elemental composition of things. You can actually get down to elements. This is why when you work with these people, they immediately tell you jokingly, but also seriously, don't lick the manuscripts because they're very poisonous, right? They have arsenic and mercury and lead and all kinds of things in them, right? Um, but these maps, obviously, they're at low spatial resolution, but these give us a lot of specificity about pigments that are in here at the elemental level, right? I can do 3D imaging again, take illuminate from four different angles and take pictures. I can get a normal map, I can get an albedo map, and then I can get to a height map, which then I can convert into a sort of 2.5D rendering of the surface. So for this particular thing, that looks like this. The Z direction here has been amplified by about a factor of 10, but you can learn a lot about how these people would make these manuscripts. They would actually take the piece of parchment and then they would score it with these lines, and that was how they would align the writing and make sure that it was really square. This one is rather odd, apparently, because it's scored from the backside, which was apparently weird. But this tells us a lot of information just about how the thing was created, the process by which they actually went through and wrote it. I can take those end members that I derived and very simply do a partially constrained linear unmixing in the hyperspectral data. And so I can go in for every pixel and tell you what the abundance is for every pixel, for what the abundance of every one of my exemplar pigments is in that pixel. And so these are some blues. Most of the blue here is lapis lazuli. These are greens, which is mostly indigo. This is red that's vermilion. There's a second red in this particular thing, but this is the vermilion one. So this is pretty straightforward. So now I can start to register all that stuff together. So this is the albedo map laid on top of the height map. And you can see it lines up really, really well. This is when I lay the XRF on top of it. So now I can really confidently tell you that these parts of the red, but not these parts of the red, were made out of vermilion. I can overlay my three abundance maps as just an RGB image where I register the per pixel abundances in red, green, and blue. And so you can see how they lay out. Iron gall ink has a tendency over time to become slightly brownish, slightly reddish. And so the algorithm confuses that a little bit and says, okay, this is not made out of vermilion, but it does have a reddish tint to it. So that's why it looks like that. This one's the interesting one. So it turns out there's actually two types of black ink in here. Um, and apparently this was a slightly uncommon but not unseen approach to how they would build these things. So there's two different people making this page in the manuscript. There's a scribe that's actually writing it, and then there's somebody that gets paid to do the illumination, right? The person that actually gets paid to do the drawing. And the guy that did the writing used iron gall ink, and it has a very distinctive signature, and so we can pull that out easily. And the guy that did the drawing, it turns out he probably used something like what we would call a pencil, although it wouldn't have looked like that, some hard piece of carbon black ink. And they actually would draw the outline of this thing first, right? And these outlines in carbon black line up very closely with indentations in the parchment that we get from the 3D image. So they almost created this thing almost like an old kid's paint by numbers thing, right? They would draw the outlines and then they would go in and they would fill it in whatever pigment they wanted to. So now by imaging this thing in multiple different modalities, fusing all the data together, lining it all up, we can actually tell a pretty good story for how this particular illumination was created. Okay, Guff map. So some of this work is kind of old. It was a PhD student of mine named Dee Bai, who now makes Google camera phones. This is what got me started. So this is the gut map. There's a good web page down here if you're interested in it. It's pretty big. It's a little over a meter long, half a meter wide. Um, uh, Maps at this time of the year or at this time in history, the top of the page pointed towards Bethlehem. So this is how you would read it, even though north is down here. Scotland, a little bit of Ireland down here, Wales, England, a little bit of Europe up here. Um, Late 14th century, they know that by looking at the town names and studying the writing. You can see it's actually two different pieces of parchment. There's a seam here. This one is lamb and this one is, or sorry, this one is sheep and this one is lamb. There's a lot of interesting things about the map. There's some weird things just because it was the Middle Ages. These are giant fish in the ocean. And there's a shipwreck here, which they think was some sort of like there was a famous Norwegian princess or something that was coming to England and her ship wrecked or something like that around this time. It's really accurate down here, right? It's really, really accurate. You can sort of make out there's a castle right here. That's London, right? And so there's a whole bunch of theories about how they made it, but essentially they don't know, right? Scotland is not particularly accurate, but at this time, most other maps, if you had to draw an island, it was just a blob, right? They had no airborne or satellite-based imaging. They had no way to map out the coastline. And so it's very unclear how they actually did this. It was probably rolled up and stood on end, and it got wet down here, which is what this is for. But it was used and modified for over 100 years, and they know that because of the place names. Interestingly enough, so the little buildings here represent individual little towns. If you had a little town, you got a little building. If your town had a little castle or something on it, you got a little building with a castle. And if you had a church, you got a little steeple. And in some of them, they had both, right? But these lines are the things that have fascinated people most about the map because they're the distances between towns, and they're super accurate down in the southeast part of London or part of England. And they don't really know how they did all that. Anyway, there's different types of red on here. Green represents water. All the writing is done in iron gall ink. Some of it has been faded away. Some of it has been intentionally blotched for various reasons, and I'll show some results of that. So one of the things DBY did was they asked us about the red inks, right? And in particular, across the entire image, were all the red inks the same, right? In other words, would it all have been made at the same time, right? Remember, pigments and inks and these sorts of things were handmade, right? And so if one person did it, they might have their own recipe for something and they might be able to repeat that recipe a lot. But if multiple people did it, there would be subtle differences. And can the hyperspectral imaging actually be able to cue off of those things? And it turns out it can. So one of the things D did is he went and he extracted all the red pigments, anything that was visually red, he pulled that out, said we're going to ignore everything. And then he did materiality sort of investigation of those red pigments to try to figure out automatically how many unique classes were there within the red pigment class. And it turns out we think there's 5. And then he said, okay, for all those 5 classes, go back and for every red pixel, tell me which one of those 5 classes it belongs to. And then he projected all that back into the map space. And you can see there's some obvious patterns here, right? So up in Scotland, most of the things are this dark blue class or maybe a little of the cyan class. All of the writing of the sort of county names and stuff is in a particular type of red. And then when you get back into here, not only does the map get a lot more dense, but there are specific regions where all the red that was used to draw the town names is a very different green than some of the other ones. This whole is London, right? And so as they started to develop their understanding of the towns and villages and potentially the trade routes that came out of London, they updated the map. And they did that using a slightly different jar of vermilion, right? So that was kind of cool. It supports the current theory of how the map was made. Catherine Delano Smith is sort of the lead historian on this project. They have this theory that around 1390, they drew the entire island and they laid down the information that they knew. About 25 years later, they chopped it off at Hadrian's Wall, which is the historic boundary with Scotland, mostly because they didn't know much about Scotland and they didn't care. And so they only worked on this part. And then as London became sort of the financial center, then they only concentrated on the area around London. And the results that we just showed seem to support that particular theory. An interesting feature on the map, though, that is still relatively unknown is if you look at the rivers that come in from the North Sea and stuff, you see a lot of these like really hard lines, really hard boundaries in the green pigments. And it turns out you see them everywhere. So this is multispectral data, but within the multispectral, again, if you know what you're looking for, you can create a simple band ratio algorithm that will go in and tell you what's the sort of likelihood or the amount of copper that's actually in that green pigment because it has a specific unique absorption feature, a very broad absorption feature around 900 nanometers. And so now we can map out where on these green pigments did they use copper and where did they use indigo. You see this on virtually all of the rivers on the map. It's unclear what they are. There's some theory that like this was an indication that would have been visually very obvious at the time and indicated something like this is where you can cross this river at low tide on horseback or something that would have been very colloquially well known to them, but is sort of lost its meaning to us. But this is sort of an easy afterthought of what we can do on the pigment mapping side that tells us, again, something about their knowledge of the area as well as what they were intending to do when they made the map. Blue. Team Pigment was formed because of blue. Blue pigments are still incredibly expensive to make. There's been some significant discoveries within the last 5 or 7 years by somebody at Oregon State or something. Completely new way to make blue pigment, right? There was a big science article about it a couple years ago. Back at the time, it was incredibly expensive. There's a couple of different ways to make blue. There's some really cheap ways, and there's a really, really expensive way. The really expensive way is to go get lapis lazuli, right? Which means you have to know where to go in Afghanistan and dig it out of the mines, because that's pretty much the only place where it comes from. These are 2 chunks of lapis that Andy has in his lab. Just for grins, I went on eBay the other day. If you want to buy a chunk of lapis like this that weighs about 15 pounds, it's like $400, right? So it's still pretty expensive. But you see it in manuscripts, right? And so that tells you if you see any blue in a manuscript, it tells you that it was expensive. And if you see lapis, it tells you it was really expensive, right? And so one of the things they were always chasing in those 300 plus manuscripts is what is the earliest recorded use of lapis lazuli in a manuscript in the UK? Because that tells you that at that point in time and any time after, there was some sort of economic commerce between Asia and the UK, right? So they currently hold the record for the earliest known of lapis in a manuscript at, I don't know, 520 or something like that. So we collect multispectral and hyperspectral imaging, and you'd like to be able to say really quickly what's lapis and what's not without having to go through some really advanced thing. It turns out lapis has some unique spectral features in the infrared that we cover with both the multispectral and the Vis-NIR-SWIR thing. And so we came up with a really simple visualization scheme. So this is a folio, an illuminated folio out of a prayer book at Utrecht Library. So this is not particularly old, 1500. You can see it's the beginning of a section or a segment. Got a big H here, drawing of the Virgin Mary and the Christ child. And then there's angels down here playing the organ and a bunch of other little things around here, right? So again, how did they actually create this drawing? What materials did they use? Well, again, we figured out this really simple way to make a false color image where in the false color image, if it's blue, it's something inexpensive like azurite. If it's magenta, it's lapis lazuli, right? And so now we can very quickly take all of the images that we have. If we see there's blue in them, we can immediately start to map out. Because we calibrate to reflectance, we know that this should always work. In this particular case, the H is probably azurite, as is most of the blue in the scroll work around here. Again, that was probably done by like, not exactly an intern, but the low-level guy, right? And then they left the space for the intricate drawing of the Virgin Mary to the person who was really an expert at this sort of thing. And that guy used lapis. This is pretty common in these religious texts that they would use the most expensive materials to highlight what they deemed were the most important people. Interestingly, you also get the angels down here playing this organ are done in lapis, and the bird up here is done in lapis. And I'm not sure exactly why they would have done that, but. Okay, faded text. I've only got a few more examples here. Text can be subvisual for lots of reasons. It can be intentionally erased. It can just age. It can be damaged due to water or all kinds of things. Blotting, there was this theory in, I think, the 1700s that if you had a manuscript or something and the ink had faded, you could take like a rag or a sponge and dip it in red wine and blot it on the manuscript, and it would increase the contrast. And it works for a little while, but then you just get a red wine stain on your object and you can't read anything. So we do a lot of work to try to get beyond that. Like I said, sometimes if it's been erased or if it's just faded, the ink particles, particularly for iron gall ink, they diffuse into the medium and then they suppress fluorescence signatures. So we do a lot of fluorescence imaging for this. Here's an example again of the Guff map. So this is Durham again. And you can see we think this is an area down here where they blotted it with wine because it's got a very specific shape. These might just be blemishes on the skin of the sheep. But anyway, we can use a combination of ultraviolet and reflectance images in a very specific way to essentially bring back a lot of that text, right? So by suppressing the signature that's the reflectance off the surface of the parchment and enhancing the ultraviolet signature, we can actually get down to text that really pulls back a lot of things that are unreadable here. Um, the historians didn't particularly care about this image because all these labels are correct and they know where it is. So I was kind of bummed, but we applied this technique to the whole map and they learned a lot. Okay, this one is kind of a funny one. The most interesting thing to me about this particular result is that there's a lot of people that appear to care. Um, in the paleography world, they're very interested in understanding the process of creating these manuscripts, and in some cases, how the materials and tools impacted what they were able to create, right? And so in some manuscripts, you can actually see as the scribe was writing, the ink was becoming either thin or different, and you can see it sort of starts to fade away. And then they would dip the pen again, and it's dark, and it would sort of start to fade away. And you can actually visually see this. So people know that, this was their process sort of thing. But you can only usually see it when the scribe was not being particularly careful to make the text look uniform. You have to remember, these things were made by hand. On a good day, a scribe could write 6 to 8 pages, right? So you're there for 8, 10 hours a day in a cold castle or something like that, working by candlelight, writing 6 pages a day for your entire life. Um, it's not visible in expensive manuscripts, but the question is, can we actually see it? Um, we can use multispectral or hyperspectral because all I'm going to do here is leverage the fact that principal components analysis, particularly when you apply it to sort of one class of material, can be very sensitive to subtle changes in that material, subtle differences in that material. So the differences in the ink could be because of the density that the pen was it was getting thinner. It could also be because you have to shake these bottles all the time. The iron stuff, the stuff that makes it black sort of settles out. And if you don't shake the bottle, then the ink just isn't as dark, right? So what we're going to do is we're going to take PCA just straight out of the box, but I'm only going to apply it to very specific parts of the manuscript. I'm either going to look at just the text blocks, or I may just filter out all the background, just pull all the pixels that are just the text. And the key here is that I don't want all that other ancillary stuff, all the reds, the blues, the greens of the illumination. I don't want that stuff to influence the statistics of the covariance matrix, which I'm then going to try to apply an eigenvalue decomposition to, right? So I don't want to mess around with that. So I calculate the rotation based only on the text, or as close as I can only on the text. And then I apply that PCA rotation to the entire image just because it's easier to do. And then we can visualize it in a false color visualization, and we can look for patterns, right? So for those of you that haven't seen these sort of things, in the images I'm going to show you, the false color images, the colors they're not intended to represent what the thing looks like visually. Things that are similar color in the false color image are likely the same material. Things that are different colors are likely different materials, right? So this is a particularly old manuscript. I think it's like 12th century or something like that in the library at Durham. So again, I just extracted all of the text and I computed the PCA transformation, and then I applied it to the entire image, and you get something that looks like this. Now, some of this is because the page is crinkled, and so you can see some of that here. But a lot of it, like down in this corner where the page is pretty uniform, you can still see color differences in the PCA, right? So this is an okay example. It's not a great example. This one, you can stare at this thing all day, and all these letters look exactly the same. But if you apply this technique to it, you get very, very distinct and regular patterns. And so this is telling you that the scribe could only write 4 or 5 different letters before they'd have to go back to the jar and re-ink their pen or shake it or do whatever they were doing, right? And so this is the pattern of how the guy was actually working, right? Write a few letters, go back, write a few letters, go back, right? And we see this in a lot of places. This one is particularly interesting. This is a religious text that's in Christ Church College at Oxford, and we went and imaged that. And you see a very distinct thing here. Now, this one is the one that surprises people because this is an extraordinarily expensive manuscript. So this was created specifically for Cardinal Wolsey. Anybody know who Cardinal Wolsey was? So Cardinal Wolsey was essentially the second in command and the second in command of the Church of England under Henry VIII. right? So they referred to him as the second king, right? So he was a super important dude. And at one point in time, they spent a ton of money building this religious book for him. And so the best scribe in the land was chosen to do this. Cardinal Wolsey, as a lot of people under Henry VIII, died because he fell out of favor and conveniently died when he was moving from, I think, York to London to be tried for treason. But we see it on a bunch of pages, and it's very similar. The other really interesting thing about this is we know who wrote this manuscript. Because it was such an important manuscript, it was actually written by this guy named Peter Megan, right? Who was sort of famous because he's called the one-eyed scribe because he only had one eye, right? So Deb and I have already decided when we retire to England and open a pub, we're going to call it the one-eyed scribe because that's just an awesome pub name. So the question now is, can we go find a bunch of other manuscripts that were written by this guy, and do we see the same pattern? And was this just his style of writing, right? Real quick, I'll just go through this quickly. We've done a bunch of other things. Lots of imaging trips to Oxford, Edinburgh, and Utrecht. Most recently, they went to Hereford, so I've been playing with that data. We've done some art conservation stuff. I can show some pictures of this if we have time. Lots of imaging sessions at Durham. Durham has a Shakespeare first folio. So the first folio was the first printed book that was the complete works of Shakespeare. They only printed, I think, like 700. There's about 200 that still exist. Durham's is particularly famous because it was stolen out of the library in 1998. And then they got it back in 2008, and it's in really, really bad shape. And so they're doing a conservation on that. And so we supported a bunch of work with the conservators to try to understand what to do with that. They also did a little documentary, and so they filmed us imaging the thing, which was kind of weird. Been to Hampton Court. This was particularly fun, right? So I was an administrator for a long, long time. And in my previous life, Python didn't exist. So I had to teach myself Python and write a bunch of code. And my PhD students thought it was hilarious because we'd meet and we'd talk about their project. Then we get to the end and I'd be like, okay, I have a Python question for you now, right? Because you all know Python and I didn't. And we did a lot of other stuff as well. So again, this isn't really rocket science, but it is very new to a lot of this community. The idea that you can do this sort of quantitative and qualitative analysis, that you can have these end-to-end workflows that are documented, robust, repeatable, all these sorts of things. If you calibrate the data up front, and so you know you've got it in reflectance, you can predict what the result will be at the end, so you can understand how to interpret it. Hyperspectral systems are still pretty rare, although the tools are, most of these people, they buy a hyperspectral imaging system. The first thing they do, they go to an NV workshop and they get completely overwhelmed. For those of you that have used NV, 99% of the stuff in there is not applicable to this problem. And so they throw up their hands and they don't really know what to do. But the goal is to always provide better information to the scholars who can understand these objects, right? And so that means we always have to be mindful of what are we actually producing? And is it something that provides value to the people that are actually going to be trying to interpret it? So what's next? So I now have a reasonably permanent appointment over there in the Institute of Medieval and Early Modern Studies, which is hilarious to me because I don't know anything about medieval and early modern studies. But all it gets me is an email address. So now they collect data with either the hyperspectral or the multispectral system. They put it up on their servers. They send me an email. I pull it, run it through the workflow, get whatever products we need out of it, push them back to the servers, and we can move on. So it's a lot faster. They just imaged the Hereford Gospels, which is an 8th century Gospels that is held at Hereford Cathedral. We first imaged that with one of our Misha systems and made some discoveries, but it wasn't quite sensitive enough and high enough resolution. So Andy went back and imaged it with his. And so we're learning a lot about that. There's this Utrecht Psalter that they imaged, which we did an initial quick look at that one. All the drawing is in iron gall ink, but they know there were multiple scribes that did it, so we're trying to find commonalities and differences across there. And then we're trying to push this into art conservation. Just this spring, I wrote a proposal to the National Endowment for the Humanities to try to really push the state of the art in hyperspectral use for art conservation. Everything you do there is nonlinear. Everything you do there is intimate mixtures. How can we train up probably some sort of AI/ML system or something that can try to undo that nonlinear transformation to go from individual pigments that are mixed together to get a very specific look at a piece of artwork? We are fortunate that we have some really strong partners, and so hopefully that proposal will get funded, and we'll know that hopefully in January-ish. We collected so much data in Durham. We have a list of like 6 papers that we have to write now, and it's now it's just a matter of finding time to do it. But that's it. Anyway, that's me. Um, Jan encouraged us to put this QR code about the Center for Imaging Science in all our talks, which I've been doing for a while. And so anyway, there it is. If you'd like to know more about the Center for Imaging Science, you can take a picture of that. And that's where I was for a year in Durham. Thanks. Roger, don't ask questions. hmm So the first folio was stolen out of the Durham Library along with a dozen other manuscripts in 1998, and it never seen from again. There's a gentleman at RIT named Steve Galbraith who is the curator of the Carey collection it's the special collection in the RIT library. Previous to here, he worked at the Folger Shakespeare Library in Washington, D.C. And he was there one day when a guy walked in with a beat up old briefcase and he put it on the front desk and he opened the briefcase and he pulled out essentially a hefty garbage bag, and in it was the first folio, and it had been ripped and torn apart. They tried to take out all the things that would have uniquely identified it as being the Durham one. He wanted to talk to an expert about it. Somebody came out of the back, and he tried to tell them the story about how he had found it in his aunt's attic in Cuba when he was cleaning out her house, and he wanted to know if it was a real Shakespeare first folio. So they said, we'll have to look at it. They took it into the back and called the FBI because they knew it was. Um, that guy went to jail in England eventually, not for stealing it, because they could never prove that he actually stole it, but for trying to sell stolen property. He went to jail, but the folio ended up back in Durham. And so when they're doing the conservation, the big question is, should they put it back together? Right? They could take the binding off of it. They have most of the pieces and parts. They could rebind it and make it turn into a book that you could actually read. And so they did a lot of focus groups. They talked to a bunch of Shakespeare scholars and people in the town of Durham, and they basically decided not to do that. As a book, it's pretty boring, right? I can go to any bookstore and buy the complete works of Shakespeare. We know what's in it, right? Nobody is actually going to sit down and read this book. The interesting thing about it is the story of how it got to the condition that it actually is. And so now the challenge is, how do they just sort of stabilize it so it doesn't get any worse, right? So that it can stay in that condition. So that's going to go on display in the Durham Library, I think in March or April. But yeah, if you're ever in the Carey collection, go talk to Steve about the day some guy showed up from Cuba with a stolen Shakespeare.